Welcome to Catch This. My name is Trudy Ray. It is well known that stress and exposure to UV radiation can reactivate replication of latent herpes simplex virus type 1 and type 2 and the painful lesions associated with these infections. But why is that? In this video, I will go over the molecular mechanism of this phenomenon. Don't go away. During a primary infection, HSV1 and HSV2 replicate within the epithelial cells of the skin and mucous membranes. Progeny viruses then travel to sensory neurons, where they remain dormant as extrachromosomal episomes for the remainder of the host organism's life. Occasional external stimuli can trigger their sporadic reactivation, potentially leading to renewed viral replication and symptomatic disease. Many cellular functions are controlled by an assortment of externally triggered regulatory mechanisms that impact when and how certain genes are expressed without changing the underlying DNA sequence. These are known as epigenetic regulations and include processes like histone methylation and phosphorylation. Histones are proteins that function as spools around which DNA is wound into units called nucleosomes, which are then further coiled and condensed into a fibrous material called chromatin. Chromatinization protects DNA from damage, keeps it compacted, and prevents it from becoming tangled. However, this extensive coiling and folding also represses the expression of genes because they cannot be accessed for transcription. The addition of methyl groups to the histones can lead to further repression of gene transcription, depending on the location of the methyl groups. Further modification of histones by addition of phosphate groups, also known as phosphorylation, can also prevent or induce the transcription of genes from that DNA, and hence regulate the expression of these genes. As it turns out, epigenetics plays a major role in the stress-induced reactivation of HSV. Latent HSV DNA is wrapped around histones that have three distinct methylation patterns, denoted as K9ME2, K9ME3, and K27ME3. This means that the methylated amino acid, lysine, labeled by its one-letter abbreviation K, can be located at position 9 or 27 of the amino acid chain in the relevant histone. Additionally, K9 can be modified with either 2 or 3 methyl groups. For example, the K9ME3 modification has 3 methyl groups attached to the lysine at position 9 of the histone. Methylated lysines can bind additional proteins, which can further add to the repressive effects of methylation. In particular, K9ME3 binds a transcriptional repressor known as heterochromatin protein 1, which blocks attachment of the factors needed to trigger a new round of replication and gene expression. A pivotal study published in 2015 clarified the molecular mechanism by which stress induces reactivation of HSV1 in mice. The authors showed that a kinase known as C-Jun N-terminal kinase, typically referred to as JUNK, is essential for reactivating HSV1. Kinases are enzymes that activate or inactivate functional molecules by phosphorylating them. The authors found that when they induce stress in mice with various drugs or stimuli, the amino acid serine, S10, located adjacent to K9ME3, was phosphorylated, and this phosphorylation appeared to prevent binding of heterochromatin protein 1 to K9ME3, thereby preventing its repressive effects. And phosphorylation of S10 was accompanied by reactivation of HSV1, as evidenced by viral replication. The authors also found that when they induced stress while inhibiting JUNK activity, S10 was not phosphorylated and reactivation was blocked. These results confirmed that JUNK-induced phosphorylation of S10 on chromatinized HSV1 DNA is a crucial process in the reactivation of HSV1. It is likely that JUNK also phosphorylates the serine adjacent to K27, meaning S28. However, further studies are needed to confirm this hypothesis and its relevance in reactivation of HSV1. It is noteworthy to mention that this mechanism was resolved in a mouse model, and it may not fully parallel what happens in humans. Nevertheless, these results provide valuable insights into the signaling pathways that appear to be key in HSV1 reactivation and thus warrant further exploration in the context of human infection. If junk does reactivate HSV1 in humans, a strategy to potentially prevent HSV1, HSV2 outbreaks in infected people could be to use drugs that block neuron-specific components of the junk signaling pathway. That's it for today. Thanks again for listening. My name is Trudy Ray, and I'll see you next time on Catch This.